the interaction and as well as learning process. Okay, so where we maybe you will be sharing the ideas as well as you gain the ideas, and you know what better way we can do the science. So overall idea is this one, and most of you as you are youngsters, you can discuss many aspects with the resource person. He is very friendly, and you will see once he starts interacting with you. So you need not to worry whether you have a small doubt or whether you have that doubt whether it is worthy to ask or anything. You need not to hesitate in any way. Okay. Totally we have 12 lecture series, that is of nearly 1 hour, and along with that even we have tutorials in between. So tutorials are mainly, they are focusing on the interactions, and maybe you can whatever the doubts you have, because yesterday only I have circulated you what about the tutorial content what we are going to discuss is today. So you can go through that one. If you have any doubts, you can get it clarified, because basically we are working on how we can use this in vitro and in vivo models, and what way we advance the science using these models. That's what our major goal is. And basically Dr. Nag, he is a meticulous planner, and he has a in-depth knowledge about the cancer, because he has worked on different models of cancer. That's why you will be benefited a lot. And his knowledge to be utilized to the maximum extent, that is with you. How best you use, it is up to you. And moreover, not be uncomfortable with this one, because it is a kind of interactive session, and you can always have a good interaction with him. Suppose if you cannot ask any questions in the session, even in tea time or any leisure time, you can approach him, and not only restrict yourself only to the cancer biology. He has a in-depth knowledge about even writing the grants, collaborative works, because even he has struggled so much to come to that much position. Because yesterday we were discussing about how he was struggling to get a small thing done at Mysore PhD. So you can learn that small tips will help you a lot. When you are doing research, sometimes you think that, oh, this is not working, that is not working. So that time you imagine how much struggle he might have done to reach this maybe the position. So you can get inspired by the thinking. That's what always I like, his way of thinking. Always he is positive about the science, as well as the overall culture. Everything has a positive attitude towards that one. So you should have certain things which you can take away from this interaction, because 5 days is a very long time. Even he has spent 5 days, means is actually for him it is nearly around 5 months time, because he has to spend that much of time. And you should make use of that one to the 100%. That's what I expect as you, the participants of this program. So out of the 12 lecture series, he will be delivering 10 lectures. Maximum chunk he will be covering, and I will be taking only 2 lectures. Basically we are telling about the basic science what we did in our laboratory, that maybe with the related to this, the in vitro and in vivo model, I will be discussing about that one. So maybe you can focus on what he is telling and what are the aspects to be covered, because every now and then he gives some hint. The way you catch, that is up to you, because each time he throws you some knowledge or some hints, just you have to catch that one. That is up to you, because as I was associated with him, he is full of ideas. So make sure that you are into this one and fully involved in this one, and let us not be restricted only to science. Let us be interact with him, and you can discuss all other aspects, how to go to maybe US. Some of you are at the verge of completing PhD. You can take all information from him. And moreover, these topics, we will try our level best to complete all the topics which is mentioned in this one. We have put a lot of effort to make sure that everything to be covered in that one. We make sure that we will actually try that our level best to cover all the topics. And the day one session, I have circulated already even the hard copies of this one, it is with you. So this formally we will start with actually the basics of exploring the mechanism of cancer progression and therapeutic response. And he starts with insight into the pancreatic cancer. So there he will be explaining you about maybe the what way the cancers can be approached, and he has his own way of the maybe interacting with you. And along with that, we have mentioned it as problem solving session. This is basically, we want it to be a kind of interactive session rather than one-sided approach, because you have certain knowledge and he has the overall idea about that particular topic. So we gave you a hint what kind of questions you can have an interaction. So it doesn't mean that you should ask that question only. Maybe related one, or whatever you find something which you have not understood, that can be discussed here. And second, after lunch, we will have basics of cancer biology. Those who are not that much familiar with the cancer biology, I think that session will help you a lot, because he is explaining tumor progression and cellular mechanism. Totally it covers almost all the mechanism of cancer, how it should be studied. And then we are actually trying to link the topic one to the other, so that let it move in a smooth fashion. Because if I am taking one topic and the other topic is covered in some other time, you may not get what is actually we are going to convey you. So that's why we have taken that tutorial session as differences in cancer type and studies. There you will have a discussion. And then, because sometimes it should not be a one-sided way of interactions. So we want to know what actually you grasp and what exactly you expect from us. Next day, that is second day, we are going for translational journey from molecular mechanism, early phase of clinical trials. Because whatever he is speaking, already he did all such things, because he practically knows the problems or maybe the bottlenecks of each and every aspect of this the work. So you can interact with him. And the session, you can see that later I will briefly explain about the study, what we did. Later he will start with tutorials again. And lunch, after lunch we are having once again one session where designing cancer model to study cancer progression. Here only actually most of us, we have the problem, what kind of model we have to design, what best way we have to do the research. So in such occasion, better you interact with him, because he knows the conditions of Indian state universities as well as the best labs in the world. So he has a long journey. So that journey will help you even to design your studies when, suppose if you are doing the research or if you are going for the research. So the third day we will slightly go ahead with our own, maybe the topic where targeting cellular signaling and tumor microenvironment, because nowadays the drugs, they fail because of the modulation in the tumor microenvironment. The cancer cells have their own atmosphere. They may prevent the drug interaction, or they may actually make the drug to enter. So these topics will make you to think in a different way. Why or what way we have to target or focus on cancer, maybe if you are working on the drug discovery part. And later on, linked to that one, maybe this is the one, maybe the second talk, what I will be telling is that how maybe the Indian scenario I will come up with, where the bioinformatics as well as the drug discovery together, we did a preliminary study which may help you to go forward with, along with the advanced study, what Dr. Nag will be telling you. And later on, the discussion will follow the models to study tumor microenvironment, because you can see that there is a link between the tutorial as well as the a topic which is covered, so that you can follow the topic easily. And later on, after lunch, he will tell you about designing therapeutic strategies in cancer research, how to design your therapeutic study, what best way, the overall therapeutic, because it is not limited to the in vitro or in vivo, or even you have to go for clinical trials, that is the phase 1, phase 2 or phase 3. So what may be the things that is associated, he will be explaining about that one. So we will try our level best, because that's what we planned meticulously. This is actually the plan of not one day, it is nearly 1 year. We planned everything. And next is that he will be telling about, because India is rich in all the natural compounds, why we selected that topic. And we have that potential natural drugs with us. What best way we can use that one for the cancer prevention, because even he has worked on various natural products. So what way he approached, what way he did the innovative thing, that he will explain you. And understanding the basics of cell culture, I will be explaining. That is an interactive session. We need not to be too formal, where we will be studying only the basic aspects of science, because with the Indian scenario, we may have certain limitations. In that limitation, what type of work we can do, that let us discuss there. And next is that he will be explaining about in vivo models in cancer studies. He has worked on various, maybe the cancer models, starting from maybe the regular mouse, what you have seen in the lab, till that GEMS, that is genetically modified animals, as well as knockout mouse or whatnot. He has worked on transgenic mice and all. So he has in-depth knowledge, because he worked on each aspect. He knows what it is. And later on, we will just have a visit to the cell culture lab. Maybe some of you may be new to cell culture lab. We cannot enter directly into cell culture lab, because active work is going on. From the outside, we can see what and all the work is going on. You can observe the cells, or you can see the arrangement of the labs. And along with that, what are the instrument facility Mangalore University has, that you can study. We have NMR, confocal microscopy, as well as even we have a very good nuclear science laboratory, that even you can think about collaborative work, because the total concept of the workshop is to maybe to enhance the collaborative kind of research. You can find out the ways where you can collaborate the things with the faculty members, or maybe the resource person, everything. And later on the 5th day, so we Professor Satish sir actually helped me to design certain timings. Actually, he suggested this is the best timing for all the participants, because some of the participants, they are coming from different places and various places. It may take some time. That's why let us stick on to 10 o'clock to start the program. So I thought even that is the best idea, because sometimes if you are stuck in the traffic, so it will be difficult for the participant to reach the program in time. That's why we have made sure that it starts at 10 o'clock. And the last day, because we know that many of you are from the out of this Mangalore city, you may find it inconvenient to reach your place if you don't wind up within time. So that's why we starting this session at 9 o'clock, and by 12:30 we should have our valedictory function, so that maybe by 1:30 you can finish off your lunch, and you can plan your journey accordingly. That's what I communicated you also once I circulated this one. And 9 o'clock he will be explaining about the compound screening. That's what most of us we usually do in cancer biology. And important thing is that he will tell you about even the targets, what should be targeted and what way we have to target the drug. And later we have a examination session. It is not a regular degree examination. Don't be that much serious, but we are supposed to assess you people, because we cannot give the certificate as it is, because 5 days you have learned so much. At least we should know what actually we taught you. So for that one we have a kind of assessment. So the assessment will be based on whatever we teach you here. We don't go beyond that one. So basically it is related with the topics that is covered in this week. And last stage, that's what actually Dr. Nag, he highlighted, because ultimately we have to make sure that overall what happened with these 5 days, that we kept it overall discussion on cancer research, models, therapeutic and interaction session. So the last session will be that one. You can have any kind of discussion, and we are not restricting anything that can be discussed here or in this platform. And I want your active participation, because even the resource person will be happy to interact with you. He has so much knowledge and ideas. So ultimately it is up to you to make use of this one to the maximum extent. Okay. With this, I told about you how the program runs. So this is way we have planned, and we made sure that it should help all those who are attending here. And whenever you find time, don't hesitate to interact with the resource person. And if you have any difficulties, let me know. So with this, I think the program will be really helpful for you. You can take the memories of this event, so that your planning of the research will change, and your way of thinking about the cancer biology and the way you think about the research will be a kind of transformed one. I think that will help you a lot. Okay. With this brief introduction, I think I should give a platform to Dr. Nag, because I will be speaking more. So I will request Dr. Anita to introduce Dr. Nag to the gathering again, so that we will have a formal interaction later. Thank you.
Thank you, sir. Distinguished subject expert, Dr. Nagaraj Nagatihalli, and dear participants. It's with immense pleasure and great honor that I extend warm welcome again to all of you to this 1 week GYAN program. And it's my privilege to introduce our distinguished guest, Dr. Nagaraj Nagatihalli, a leading expert in cancer biology and translational medicine. Dr. Nagatihalli currently serves as an associate professor of surgery and graduate faculty in cancer biology at the University of Miami, Miller School of Medicine. He is also an integral member of Sylvester Comprehensive Cancer Center, where he leads groundbreaking research on pancreatic, lung, and oral cancer. Dr. Nagatihalli holds a PhD in biochemistry from DFRL Mysore, following which he pursued his postdoctoral research at the University of Louisville and Vanderbilt University. His academic journey has been marked by a commitment to unraveling the molecular mechanism underlying tumorigenesis, drug resistance, and targeted therapies. Over the years, Dr. Nagatihalli's work has earned numerous national and international recognitions. Some of his most notable honors include Young Investigator Award from the American Pancreatic Association, Stanley Glacier Research Award, University of Miami in 2016, James Esther King Biomedical Research Award from Florida Department of Health, multiple travel scholarships from organizations such as National Pancreas Foundation, the American Association of Cancer Research, and Collaborative Alliance for Pancreatic Education and Research. Dr. Nagatihalli has been highly successful in securing competitive research grants, which includes NIH L01 grants worth rupees $1.7 million, which is around 13 crores in Indian rupees, and Florida Health Research grants worth rupees $0.58 million, again around 4 crores in Indian rupees, and again NIHRO in grants worth rupees $2.48 million, which is equal to 21 crores in Indian rupees. So his research has also contributed to phase 1 and phase 2 trials, clinical trials targeting key pathways in pancreatic cancer research. So Dr. Nagatihalli has published extensively in high impactful journals with over 60 peer-reviewed research articles, 4 book chapters and 127 papers presented in various conferences. He contributed for various editorial boards such as managing editor for Nutrition Journal, associate editor in Frontiers in Gastroenterology, associate editor of Frontiers in Oncology, editorial board member for Journal of Biological Sciences, and many more. He is also reviewer for various national and international research grant proposals. Beyond research, Dr. Nagatihalli is dedicated mentor and educator, having guided over 15 postdoctoral fellows, numerous graduate students, and medical residents. His mentorship has helped shape the next generations of research, many of whom have secured independent faculty positions. Today, we are privileged to hear from Dr. Nagatihalli, who will share his expertise on cancer signaling, network tumor microenvironments, and novel therapeutic strategies. Please join me in extending a warm welcome to Dr. Nagatihalli, Nagaraj Nagatihalli. Sir, the stage is open for your delightful talk.
Hi everyone. Hope you all heard everything. Don't worry about whatever they said. I think important. I think learning, right? The learning. Whatever Joshi was saying and Ma'am was saying. Um, it's not about knowledge. Knowledge gained by doing it, doing the experiments, conducting experiments, and this knowledge is from the experience. So I'm going to share everything what we have been through many years, and I've been through many years. Just not the pancreatic cancer. I'm a pancreatic cancer, researcher. It's not on. I will on and then I will say. Okay. I don't want anything to go like. Um, it's also about lung cancer. I was a postdoctoral working in head and neck cancer, oral cancer, and then I shifted to lung cancer and colon cancer. I was not knowing when I go to United States. You don't know until certain years what exactly you want to do. It takes time. So that we will talk more if you are all interested. Why it takes more time story about what you can do, what you can learn, and what you can conduct with the facility that you have in your university or your institution in real world. Let's be practical and learn from it. And rather than, so many things that I'm going to show you that with the experience, with the laboratory, what we have conducted research, I'm going to show you. I don't tell you the story about Louis Pasteur or something else or someone else. No, we talk about what we have done, what we can do. That's all. Okay, this next 5 days, there is no history. It's only practical. This is okay, whether you can do it or not. I'll tell you whether you can do it or not. Okay, whatever they have said, it's true, but in a way that most important thing, what they don't know is that what we have been through, what I have been through, right? That we're going to share, that we're going to discuss so that you learn from it. You don't have to waste many years. So you can actually straight away just start getting the product out of it, right? If you learn something. But especially this talk, it's okay. If it is a little bit over than what you thought, it's okay. But I'll also explain to you in a way that if you don't understand, it's fine. Okay, don't worry about it. We will, next 4 or 5 days, whatever, I'm going to take one example from here, and I'll explain to you why I presented that, why I showed you the little work that I'm showing you today. And I'll explain again why I want to explain, because as a research, I will show you the whole thing, but when it comes to understanding, I have to explain to you, right? Some research probably you might have not done, or you might have done, probably you might have not noticed, things like that. So that's all. Let's do today. First talk is on pancreatic cancer, but it's a story, okay, the research story. So we in between, I will explain to you why we have conducted these things so that you learn from it. Okay, you turn on and then.
So you can hear me, right? Is it too loud? Too loud, right? I don't know how to reduce it. Now it's okay? Now it's better, I think, right? Now it's better? Now it's better, okay, good, thank you. Okay. Okay, once again, thanks to Gian for this grant. Thanks to Joshi for writing this grant together with me. So, and also Mangalore University for this, the great opportunity, support, especially, the whole Mangalore University team. Once again, I sincerely thank the Vice Chancellor, today's the program, and all the facilities they have given to all the participants and accommodation and everything, whatever they have done to make sure that you're here. Today is the first day. First talk is all just about the introducing the pancreatic cancer. So, as I said before, that I've learned the lung cancer. I've done postdoctoral in lung, head and neck cancer, oral cancer, and researched on lung cancer and the colon cancer. And I went to pancreatic cancer because one time I met a surgeon who was actually a pancreatic cancer researcher. He was not a researcher, he was a surgeon. When I met him, and he was telling me about the story in Vanderbilt University. Vanderbilt University is called Southern Harvard. Means that in America, if you take the southern part of it, okay, Vanderbilt University is considered a south region. In that south region is one of the Ivy League university. So, so we called Southern Harvard. That university, I went to that university from Louisville. The reason I went to university, I was passionate to conduct cancer research. So from in Louisville University, my mentor was a German who actually molecular biologist, and I wanted to learn more of translational studies. So I went to Vanderbilt University, joined, and when I met this surgeon, he told me about the pancreatic cancer patient stories. Like people are dying, at that time in 2007, 2008, after my postdoc, I was an instructor at that time, and when he told me the stories, I was like, what we can, I think we can do research. Then he said, I don't know anyone, so I can do it. We can start the lab. So he was excited to start the lab. He was a surgeon. He had all the ideas about the translational studies. Means that he knows patients, he knows what's going on. What I wanted is that exactly. So I was a cancer biologist. I studied in head and neck cancer. I was studied in lung cancer, and at that time I was doing the lung cancer. I never experienced the patient. I was like you all. You're studying the cancer biology, doing some cell cultures, doing some molecular biology, but I don't know where these studies were going. I was not knowing. So, but that is not what I wanted. That's not why I went to America, leaving the DRDO job, right? It's a big thing. So I wanted to really learn cancer biology. So he gave me the opportunity to start the pancreatic cancer lab. I started from scratch with the 2 cell lines, the pancreatic cancer lab, just on one table. There was no sitting table. There was no computer. There was nothing. You think that you have a problem? No, I have a problem too. When I started in 2007, there was no computer because we didn't have money. But only we had is a passion. We had the university name tagline, right? That's enough. When you have a university name like Mangalore University or Bangalore University or Coimbatore University, you have a tagline, Coimbatore University. So you take the tagline, doesn't matter where you are living, important is that where you are from. I'm from Vanderbilt University. Oh, then Vanderbilt University, oh, okay, there are people recognize that university. Okay, that means that I can collaborate and I can interact and I can get it done. There are many ways that we can interact and talk about it later on. Pancreatic cancer is one of the top 10 most common cancers. It's very aggressive, and in terms of mortality, it's ranking just behind the lung and colon cancer. Despite it's a tough disease, survival rates have improved from 5% to 13%, as I told you before, giving us more hope. However, there are challenges remain in this study. Because it's a late diagnosis and also less effective treatments. In pancreatic cancer, always a patient come to a clinic, they're all already late. Okay, because they won't know. It's a very small organ. It's hard to know do you have a pancreatic cancer or not, right? So that's why it's called late diagnosis. They're already dying, almost, right? And also there is no effective treatment. Pancreatic cancer, the reason I joined the pancreas group, the reason is there was no treatment at that time. Many people died. Many people died during that time. Now there is a hope. By 2030, it's expected to be the second leading cancer death. It means that there are more people dying by 2030. That means the expectation, the way that it is going, there are more people are dying. We will talk about why later on, right? And the survival chances vary depending on how early the cancer is found, with the rates dropping as low as 3% in advanced stages. Means that when you take different stages of pancreatic cancer, when you go to the advanced stage, there are more people. There is, survival rate is very, very low because they'll definitely die. They know that. When the patient come to a clinic, already they know, the doctor knows, they're not going to survive. This patient is going to die. But if you take a lung cancer and the breast cancer and the prostate cancer and a lot of other cancer, people survive completely 100%. Breast cancer, 100% they can survive. So because the treatment advanced, the reason it is advanced, because they poured a lot of money, a lot of researchers went into the study breast cancer long ago. Lung cancer also the same way. Big people can survive, right? So it's the same way. The pancreatic cancer at that time, there was no money, there was no researchers, there was no investment. So people were dying. So then the government has to fund, the government has to put money. It's the same thing here. The cancer is increasing. The government is now awakened. Government is actually telling, you can see that the present government is actually putting more, interest in cancer research. Okay, the survival rate, chances are, vary. As I said, in advanced stages, 3%. Despite these challenges, there is ongoing research, new studies, especially early detection. They have started early detection in pancreatic cancer. Now we have some hope. There is a blood test you can do, and there is already you can see the improving in survival in pancreatic cancer because of the investment they are putting. So my lab focuses on three major areas in pancreatic cancer hallmarks. One, there is over 90% of these tumors have KRAS mutation. The pancreatic cancer have more than 90% is KRAS mutation. We talk about these mutations in a while. Today and in next 4-5 days, we keep talking about KRAS, P53, SMAD4, all these mutations. You also know from the from a doctor, oh yeah, this patient has a mutation, this has this mutation, EGFR or whatever they call it. But another thing that you need to know, each cancer have a different mutations, a different problem, right? You have breast cancer, different problem, lung cancer, different problem. But that's why we have to understand each cancer. You cannot jump because there is a money from pancreatic cancer to oral cancer and lung cancer. But if you want to study one cancer, you have to study in depth, in detail, understand the biology before you treat. That's what I was saying. Understand the biology before you treat. And our previous studies have shown that desmoplasia, that is called stroma, is a big barrier in effective drug delivery. We are exploring ways how to overcome this barrier. The problem in the pancreatic cancer is one of the hard tumor. Hard tumor means that any drug you give it to the patient, that is not going to the pancreatic tumor. It's going somewhere else, but not, because it's very hard, because there is a lot of stroma in it. It means that fibrous, it's a fibrous tissue. It's a hard tumor. Pancreas, if you actually see, very delicate organ. But when it comes to tumor, it's a very hard tumor. That's why it's challenging for the doctor, for the researchers to study pancreatic cancer, right? And there is another one, we have established a few studies to show how these tumors weaken the immune system, help themselves grow. Remember, earlier we used to talk about just the pancreas cells, pancreas, intrinsic signaling. We always talk about this signaling, that signaling in the pancreatic cell, in one cell. Now there are so many cells. We understand that as we research increasing, we understand now T cells, fibroblast cells, stromal cells, calf cells, you name it. There is another week, the next day, probably there is one more cell will come. You keep on researching, there is one more cells will come. You get confused. But what do I do? Don't get confused. Study what you think is important, according to you. It's all important, okay? So don't worry about what others are doing. It's okay. But think within your knowledge what you can do, how you can establish. I was talking about immune system, immunology, immune cells. Very important now. Everybody talks about immunology. Everybody talks about immune cells. Studying is also, technologies are coming, and it's hard to study because there are so many technologies. Which one to use? But in your facility, in your university, again I'm coming, in your research, it doesn't matter. If you want to study something, right, you can always study. It's not hard at all. You want to study immunology, you can study with immunology without any technology. But important is that you need to awaken, you need to have a confidence within you that start doing something. You will find out one day why you cannot, right? You have to start somewhere, okay? That's important. You have to start. There is always a problem for everything, but you have to start, okay? Today, I'll show you our research work in these three specific areas of pancreatic cancer, right? Finding and targeting new cancer-causing factors. It is a risk factors. We can talk about tobacco, alcohol, KRAS, P53. We all talk about, okay, well, there are questionnaires I've given. So we talk about Indian scenario. What are the factors? One of the factors is tobacco. Is increasing here, right? When compared to Western culture. Alcohol. Can you imagine how much people are drinking now? It's too much, right? I mean, it's increasing. When compared to, you go back to 5 years and 10 years, now people are like, everybody's drinking, right? So we have to talk about that aspects too. And in a while, then I'll start talking about alcohol. Increasing drug delivery by remodeling the stroma. We also study how to remodel the stroma. How do we, okay, first you have to understand the stroma. What is stroma? Why stroma is coming, how it is coming, and then we have to target the stroma. Target means what? Can we kill the stroma? Or can we just disturb and just let we go? Like for example, there are many people crowded, but we still pass through. But how do we pass through? We say, excuse me, excuse me, excuse me, you just pass through, right? So we have to make some drug which actually says, excuse me, so that the drug will go through. You know what I'm saying? We need the drug which actually can go through that, problem, right? So we have to find, how do we find the drug? That's what I think we are looking into. We are looking into, I'm not telling you in detail exactly how we are looking into, but you have to, exactly as Vice Chancellor was saying, you have to interact with other department, understand the physics, understand something else, chemistry or some technology or some different model system, right? Then only you can actually find a way how to make this drug enter through that stroma, enter through that hard core area, right? Also, overcoming therapeutic resistance by modulating immune system. So everybody was talking about, hey, this is a cancer cell, and we can target. Now I'm going to start thinking about immune cells. Why can't I start doing something to do immune cells so that immune cells can take care of the resistance of the drug? You know what I'm saying? There may be immune cells because people don't know. We all think that, oh, probably I'm starting a research. I'm starting to understand the cancer biology. I'm maybe already everything is done. Maybe I don't know what to do because everything looks like everything has been done. I don't think I understand this. Yes, everything is done, but still you can do something. Okay? Still you can do something. But you should not worry, oh, everything is done, so you don't have anything else to do. No. There is always a way if you want to do, right? So we found immune cells, immune microenvironment, and then we targeted immune environment. Then we saw, oh my God, why we didn't learn this 10 years ago? We would have treated this patient much better, right? That is the knowledge, right? Somebody says, oh, virology, no, I don't want to do virology. Maybe you may have to do virology. You never know. I don't want to do bacteria. No, you may have to do bacteria. There's a good bacteria, bad bacteria. Okay? What bad bacteria do and good bacteria do, right? Then if you can understand and differentiate this, then you can find what is your problem in your research, whether you want the good bacteria or the bad bacteria. Then how these bad bacteria work and the good bacteria work? Like this. Start thinking and converse with your colleague, and maybe one day you'll find it. It's not a big deal. You all find something new, right? So during the progression of pancreatic cancer, several key biological events mark this aggressiveness, right? So one is the acinar cells undergo reprogramming into a ductal-like structure due to inflammatory signal and initiate the precancerous pancreatic neoplasia cells, and which leading to pancreatic cancer. Don't worry. Something that I'm giving you, you may not know. Don't worry. Every cancer starts with, like this is acinar cells here. In the pancreatic cancer, it is acinar cells. In the breast cancer, something else. Colon cancer, something else. But everything has to start from there. And then it will lead to somewhere here. Every cancer lead to somewhere in the middle. But we call it different name. Okay? And then it will go to a cancer we call breast cancer, lung cancer, colon cancer. But the technology, the knowledge you're going to gain, it doesn't matter which cancer, it's all same. Okay? It's all the same process. Only thing, I talk about KRAS today, right now. Probably when you take a colon cancer, you talk about SMAD4 mutation. You take a breast cancer, you talk about ARB. Okay? That's all the difference. But you can do any cancer, doesn't matter what. You're interested in the prostate cancer, you can do it. But we call differently. we understand the biology almost same. But there are some cancers, biology is same. There are some cancer, biology is different. But to find out, okay, you need to have some interaction, some knowledge, and also interact with other cancer people. Okay, then you learn something else. Oh, in this cancer, the one now I'm doing, maybe it is not the same way, or maybe it's the same way. We all understand, oh, okay, what do the stem cells do in your cancer? Oh, stem cells do in my cancer this. Maybe in my cancer it doesn't do that way. So these are all the understanding, these are all the biology. That's what I'm trying to tell you. Okay, and over 90% of pancreatic cancers have KRAS mutation. Over 90%. Okay, KRAS mutation is ultimate in pancreatic cancer, but it is not true in other cancer. In lung cancer, it's only 40-45. So in different cancer, different things. Remember, that's why I was telling you, each cancer is different. Biology kind of same, but if you can understand one cancer, you can start thinking about other cancer the same way. Philosophy is same, but the knowledge, the gain in each cancer, mutation, other factors involved, they're all different. Smoking affects lung cancer more than other cancer, but it also affects prostate cancer, it also affects pancreatic, it also affects brain, it also affects colon, but in a different level. But it does. It's that way I'm talking about. It can affect a lot of organs, but it affects some organs more, some organs less. You may say, again I'm coming, okay, I'm smoking from last 25 years, I don't have any cancer. Yes, sir, you don't have any cancer. But your friend or someone whom you know, he smoked maybe 3 years, but he got cancer. Why? Because there are mutations. Okay, that's what I'm going to talk about that in a while, that there are some mutations in his body or her body that is not there in your body, or your factors, your body metabolic or rate or metabolism or gene system is entirely different than her or his body. That's why you're not getting it, but that person is getting it. How do you know? You don't know. You better don't do it rather than going ahead and then worrying about. Alcohol is the same way. I'm going to tell you the story all about alcohol. Oh, I'm drinking so many years. My dad, if he don't drink alcohol, he's not going to be a human being. He has to drink. Okay, nothing happens to him. He was like, he was drinking 95 years, he's fine. He's fine. But you will not be fine. You never know your body system what it is until unless it happens to you, right? It's that way. So around 50 to 75% of the pancreatic cancer have a P53 mutation. This is another mutation. We talk about these things tomorrow or later today, a little more in detail, but at least I'm giving you a kind of hint for you guys what's coming so that you'll understand, right? That this KRAS and the P53 mutations are key drivers of pancreatic cancer. The same way, different cancers have different drivers and they have a different way of doing things. That way. From these studies, we understood that KRAS and P53 are key drivers in pancreatic cancer. Inflammation supports their oncogenic impact. What does that mean? For example, you have a mutation, you have inflammation. Bad. Inflammation to your body is a bad thing. That's why we say, oh, you drink green tea, everything will be fine. Okay, yes, you're right. You take turmeric, everything will be fine. You know what? They are all anti-inflammatory agents. Your body, if you have any inflammation in your body, anti-inflammatory agents always helps, always do good thing. Okay, that's why we don't get stomach cancer much, because we always take turmeric. Turmeric is one of the anti-inflammatory, well known in the whole world. We Indians have done a patent and everything, right? And in the same way, in the Chinese green tea, it's the same thing, same concept. But they do drink. When you go to doctor, if anything happens to you, if you go like, I have an allergy problem, doctor says, hey, you can do the allergy test, but you should not drink green tea, you should not take turmeric, you should not take this, because they're all anti-inflammatory. Okay, that's how you know that they're all anti-inflammatory. It can, help you. So, but can coffee do the thing? No. Coffee is not anti-inflammatory. So, it's just that, okay, if you drink coffee, too much of coffee, what happens? I don't know, you're damaging something somewhere. Maybe less is better, right? Like that. The molecular analysis of pancreatic intraepithelial neoplasia, that means that pannins we say here, this is a normal cell, and the pannin 1, 2, 3, and then the cancer. And this is in pancreas. The same way you go take a breast cancer, you take a lung cancer, same biology, but but naming is different. Here pannin, they are different, something else, the same thing. But in the progression, in the pancreatic cancer, it starts from normal cells to a pannin. It's an early neoplasia. The cells looks like that, and then it will go to pannin 2. Then the cells looks like that, and then the pannin 3. We know that what happens to pannin 3, and I can go in detail if you want to learn pancreatic cancer. I can tell you hours and hours, but that's not important today, right? Important is that this can happen. How do we study? What is the model? I think that we stick on to that. And when it comes to cancer, there are no, these type cells are all gone. This has become completely stroma. Now all the cancer cells are gone. Even the normal cell ductal cells are gone. You know why? Because there is no room. There is all the bad things are coming in. The stroma is a bad thing. It's all coming in. That's why the pancreatic cancer will become a hard tumor, very hard tumor, because there are no cells. If you have a cells, there's your tumors are very, delicate and not stiffened. But once the stroma comes in, it's a hard tumor. Okay. In a mouse model, we studied that this is in the pancreatic cancer mouse model. When we say KC, is a KRAS only. There is only KRAS mutation. We developed this mouse model. Don't worry about this mouse model. Each mouse model, when we talk about mouse model, right? We call in the pancreatic cancer we say CRE, specific to the pancreas. We say PTF1A CRE or PDX1A CRE. The same way. Don't worry about those names. But what I'm trying to tell you is that if I want to study lung, I have a CRE mice specific to lung cells. We know where to target lung cells. Each cell is a different type. Okay, they all look the same. They all functionally same, but they are different in some certain things. Right? That's how the investigators, scientists have investigated some markers so that these are all lung, these are all colon, these are all CRE targeted to colon, CRE targeted to pancreas. So that way we have targeted CRE. This is PTF1A CRE or the PDX1A CRE. So only we tag that to the pancreas, and then we add something else. Like for example, we add KRAS mutant cells, mutant mice, or p53 mutation suppressor gene. We add it to that and study. So if you have these genes here, like an activated or suppressed, now whether this mice makes a tumor or not. The same way, you take breast, lung, colon, anything, doesn't matter what. Study the same way, same concept, same idea. Everybody study the same way. But as I said, biology is same. There are some markers different. Mutation is different. The way we make them model is all technologically it's the same thing. Okay. But in here, in the pancreas, we have a KC means KRAS mutation. KPC means that KRAS and also p53 mutation. So in the KC, if you see, there are some ductal cells here. Ductal cells means that next stage of cancer. But this KC, if you have a KRAS mutation, it doesn't make cancer. Okay? But when you add a p53 mutation here, it makes cancer. Okay? So, sir, I have a KRAS mutation, I checked. However, I don't know how you check, but I checked. Do I get cancer? Maybe not. I'm smoking, you may get it. I'm drinking, you may get it. But if you don't have any other problem, just you have a KRAS mutation, people live with KRAS mutation lifelong. If you are taking a good diet, good food. If you don't have any other bad habits, right? I'm going to tell you the story right now. Okay? So KRAS mutations activate key signaling pathways such as PA3 kinase, ERK, P38, STAT3, and mTOR. These are the signaling pathways that I'm talking about, which we worked in our laboratory. There are many signaling pathways I'm not talking about, okay? But I'm just giving you a hint on the idea, that's all, right? There may be other pathways too. People have worked on ERK pathway. There are a lot of different EGF or GPCR of signaling. You guys may be working on some other signaling pathway. So there are many pathways, but these are the pathway in our laboratory we worked on. The reason we worked on, just a second. Hello? 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 So do you know where is he? Hello? 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 I just want to make sure that I'm recording, otherwise they may ask me to talk again. uh, hello? Sorry about that, guys. So just want to make sure. Okay, I think we will continue. The I was talking about the signaling pathway. These pathways are prime targets for the therapeutic intervention in pancreatic cancer. So it's also well established in the field that persistent acute or chronic inflammation together with the KRAS mutation significantly increases the cancer risk, particularly with the inflammatory pancreatic tumors known for this aggressive nature. So I'm going to show you here with the KRAS mutation. I just mentioned about KRAS mutation, remember? With the acinar cells, there were KRAS mutations here. And now you add the inflammation here, it can lead to cancer, right? So this is kind of, other people have, investigators have studied this, but in different way, differently. It's not working. It's not working. Hello? 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 Hello?
Hello, hello. Hello, hello. Hello, hello. Hello, hello, hello. Hello, hello. Hello, hello, hello. Hello, hello, hello. Hello, hello, hello. Hello, hello, hello. Check, 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 check. check, check, check. Check, check. Check, check. Hello, hello. Hello, hello, hello. Hello, hello.
Okay. Split. Why? Okay.
But you can't move around.
Huh?
You can't move around.
No, I'm not moving. Yeah, it should be fine. Hello, hello. Yeah, that's fine. I'm not moving around, so I don't think we need that. Huh? Hello, hello, hello. Yeah, fine. So I was talking about the acinar to ductal metaplasia. So acinar cells to ADM, that means that acinar cells will convert into a ductal type. Like you see, the shape of the cells from here to here different, right? That's how the shape of the cells changes. We say ductal type. And then the shape of the cells change again. It will become something a cuboidal kind of. So it's called pancreatic interepithelial cells. And then the shape from here to here changes to kind of entirely different. These cells are very aggressive cells. They divide faster. They increase the cell number so fast. So the reason is they mutated. They got mutated during this process. These cells got mutated. There are a lot of mutation, there are a lot of other things, maybe inflammation, maybe something else is happening to these cells. That's why they're changing, right? That's why. So the question is here that without the inflammation, without this inflammation, whether this acinar cells with just the KRAS mutation, can this become cancer? No. So far, whoever did the model, and doesn't matter what type of model they've done, they have not seen it is becoming cancer, right? Something else has to happen. Something else has to come to your body. So it can be from the food, it can be from the mutation, it can be from some other inflammation. Something has to happen, then only it will become. So this has been proved by many scientists in many models. It doesn't matter which cancer, it's the same story. But remember, again, the KRAS I'm studying and showing in pancreas that the same thing is different. Same methodology, maybe mutation is a different mutation in different cancer. Methodology, the idea is all same. The idea, whether these cells will become cancer, the story is the same thing. Okay? So the progression from the pre-malignant, or we can say acinar cells in the pancreas, conditions to cancer remains incompletely understood. Even though we think always, oh, we understood the story, always something else will come in. Someone will say, no, we have this type of cells, we should study, because we saw in this type of cells, there is always, scientists wanted to investigate something new. Everybody wants something new, right? Even the grant you write, they always say, okay, what is the novelty? When you send a paper, they'll say, what is the novelty? Because the novelty, they're asking you to investigate something new. We already know this. What is that you're going to do? What is that you're going to find out? You say someday, you're sitting there and you're seeing the cells and you're seeing something, patient tissue or anything. You're seeing something, but you saw something. It may be contamination, but you saw something. And then you start discussing with your colleague, oh, I saw something. Many times in our experience, nothing has happened. It's a contamination. It's something wrong that I have done, or someone else has done, or someone else has poured something or done something. But it's the curiosity that you develop to know what happened is what I think is going to take you to the next level, right? You have to develop the curiosity. It doesn't matter what. It doesn't matter what, whether you're doing the research, whether you're doing something else, or teaching, or you running the bullock cart, whatever. But you have to have that curiosity in your work. Then only you can do something in your life, right? It's the same thing here. The cancer biology studying, you have to develop curiosity, especially when you're doing the research. The curiosity is the ultimate for doing this, okay? And I'm just showing the critical knowledge gap in the pancreatic cancer is understanding the basic biology of it. It doesn't matter how many years we are studying. I said I entered in 2007, I'm still studying. There are a lot of gap, a lot of things that we think we know, we don't know, right? And we think that we are almost there, we are not there. So this happens. That's why we want more people to study. That's why government is spending more money. But you have to find out something new, unique, your own, and always studying, understanding, and discussing with your other friends and the scientists from different universities, some pioneer, some small things, some small hit can change the way you think, how you want to do research or study or teach. Teaching also. If you want to become a teacher, one hit from someone or something, they say, can change the teaching also same way. So the concepts are same, but the design that you're going to do is different. Okay, we published a paper on alcoholic pancreatitis. Okay, so alcoholic pancreatitis means that you drink alcohol, binge drinking we can say, or completely every day you drink, and there will be too much of drinking, and there is a high risk in India also, pancreatitis, pain in the pancreas. They go to hospital, emergency, pain in the pancreas. Some people will have a lot of pain and they die because they cannot resist, because they don't have a hospital around, they die. But like in a village side, someone is drinking too much, they die with the pain. Okay, they have too much of pain, they drink more, but they know that it's because of alcohol they're getting the pain, but they keep drinking and they get more pain. But the pain is not going to go away, but actually their brain is going away. That's why they think that the pain is still there in the pancreas. But in where the hospitals are in emergency, people go and get the treatment. All they give is steroids and stuff, because the pain is hard in pancreas. Pain is really hard. Pain is bad in pancreatitis pain. Okay, alcoholic pancreatitis is one of the worst thing. Okay, so we developed the model to study alcohol. When the important thing is this: why we studied alcoholic pancreatitis, right? You may be asking, okay, maybe he's interested in alcohol, that's why he's studying. No, of course I'm interested in alcohol study, but more important than that is that one day I was actually, one of my surgeon was telling me that, I see these patients, they have a pancreatitis pain. They all come to hospital, but I don't see any pancreatic cancer in these patients. They all be fine. They give some steroids and other drug, and they all be fine. But I asked him, okay, if they have a pain in the pancreatitis, if they keep getting the pain, they keep drinking, what will happen? Is that anything changes, mutation or anything happens to this patient if they keep drinking so much? And whether they become, any cancer patient have a pancreatitis also? Then he said, there are like 2-3% of patients that come to hospital, it is increasing, and they do have cancer. Means that when we took out the patient tissue, pancreas tumor, because when the pancreatitis is happening, the doctor cannot take the pancreas out. Okay, they only treat with externally. But if you have a cancer, they can treat. They can open they can take the tissue out, but those tissues in the side by side, he had a pancreatitis and also pancreatic cancer. That means that these patients had pancreatitis, inflammation, that means, and also pancreatic cancer, means that this patient had a pancreatitis. That means for me, it was curiosity is that, okay, why, how? To study that, I started studying that you cannot just jump onto something and study everything. Remember, it's a research. You have to study slowly. It takes years. You have to cool down and calm and patience. You have to more patience in research. So we have to study first what happens to just the alcohol. Then you study the model, you make the model, then you study, and then go to slowly next, and one day you study everything, right? So this is how we did. We studied just the alcohol. Okay, if we give this mice a lot of alcohol every day, and what happens? They enjoyed it. After a while, we saw what is the problem. And remember, there's a curious thing in this study. You will know soon that when we give the alcohol, these mice, after a while, had inflammation, it came back. You leave don't give the alcohol, they come back. Means that they're all fine. And they have inflammation, they're all fine afterwards. Means that if you continuously drink, there is a problem. But if you stop and again start, it's okay. According to the model, I'm not saying that you have to do that. What I'm saying is that according to our model, it's true that it can recover back. Means any cells in our body, it doesn't matter which cell, breast cells or the lung cells or brain cells or the prostate, any cells, right? They have a recovery stage. They can recover, doesn't matter what damage. That's how the beauty of our body. It can recover any damage. But certain stage, after it reaches one stage, that actually damage occurring every time episode is happening and again, probably not, right? I'm going to tell you that story also. Then we developed the curiosity. The next, we published that, the alcohol pancreatitis model, and we studied and we targeted. There is a story, I'll tell you not today, and I think tomorrow or day after, the whole story of that in a different way. But for us, the curiosity is that, okay, now in this model, I'm going to add KRAS mutation to these mice. What happens? Whether they develop cancer or not. That's a curiosity for us, right? Then, before that, we have to understand the biology. Okay, what happens to this? Okay, go ahead. Completely. Okay. No. So we'll talk about that ROS. We can say reactive oxygen species, how it works, and we can talk more and more. But you can completely stop it. Nothing will, it will recover back. So, but remember, it all depends on the damage. How much damage you have, or damage occurred to the body, right? It's about that. We'll talk about that more in detail. Okay. The studies have shown that pancreatic acinar cells can de-differentiate into duct-like states with injury or activation of this KRAS. With inflammation, further it drives to driving this process. So the acinar cells are ductal shape, but with the KRAS mutation, it actually forms this ductal shape. These de-differentiated cells can revert back, okay, to acinar cells after injury resolves. Means that you just leave it, no alcohol, it can actually reverse it back. It's not just the alcohol I'm just giving you one of the thing is alcohol, but it can be any inflammation. Okay, it can be from diet, it can be from the bacteria, it can be from something viral inflammation. It's the same thing. Inflammation is the same thing. So I'm just giving you one external risk factor I'm giving you, right? These de-differentiated cells can go back to acinar cells if you don't have a KRAS mutation, right? If you have a KRAS mutation, it will actually further goes further. So to study this, what we did, we did that cell specific, that is a PTF1A CRE mice specific to these acinar cells. We call CRE. We developed this mice model, and with and without the activated KRAS in this mice. So we use this PTF1A Cree mice without the KRAS mutation as a baseline model. We developed first baseline model and we studied, and whatever story item I was telling you, we actually modeled that story to study further, make sure this is how it is happening. And right, we developed the in vivo alcoholic chronic pancreatitis model by giving the 5% ethanol. It's a diet, it's a liquid diet. It's exactly, the NIH made a rule exactly how we have to treat these mice and give these mice alcohol. And any study you do, you have to have a standardized way of doing the research according to the whatever the rules and regulations, right? And the mice have more rules than human being. They can take the human being and put it in a emergency, or you can cut and open and everything. Mice, we cannot. We have to have the protocol. We have to have the approved protocol from so many organizations, making sure that killing these mice is not affecting something. So, but we can kill human being, but we cannot kill mice. So this is the rule. This is the rule everywhere. For us, the problem is, even though I have to kill one mice, I have to go through so many procedures. But patients, they can die and they can cut open. It's much easier. That's how the world is. So after we studied this histology, the whole thing histology. We don't worry about the details of histology. What we did, what markers and all. When you are studying the cancer, then you will understand that based on what cancer you are studying, what markers to study, those kind of things you will understand when you are doing the modeling. So here, in the 3-day recovery, is just the 3-day, means that you hit with alcohol, like 2 to 3 weeks of alcohol and continuous alcohol, and after that you stop, right? And within 1 or 2 to 3 days, it will be, you see a lot of fibrosis, a lot of some of these ductal cells and CK19 and all. But after 21 days, just you leave it up to 21 days, everything reverses back, just like normal. Nothing happened, as if like nothing has happened to these mice. Okay, it reverses back. So this is just showing the basic model. So also, we as researchers, we are curious, right? Just to understand more biology of it. So now I'm more curious how this is happening. That's the curiosity you develop. And this ductal type of cells, this is the 3-day. We saw here, you see the red one, red is a CK19, that's a marker for the ductal cells. We saw all this red one on the acinor cells. Look how beautifully this went back, all gone by 21 day. So that curiosity, right? Now you'll be even more curious. After seeing this, we are more curious. Okay, then what is biology within those cells type? Means what signaling is activated, what signaling is going away. You're more interested, curious to know even more and more. That's how you'll never go back and eat. The reason is that, okay, you ask postdoc, okay, how do we do next? How do we do? What do we do? How do these signals work? And what signals we have activated here and deactivated again? Those curiosity. So so next we as previously mentioned, we developed the KRAS mutant mice. Okay, we with ACNR specific Cree is actually developed the ACNR to ductal reprogramming with this in the model. So this ACP induction in KC mice, in the KC mice, I'm talking about KRAS mutant mice with the alcohol. That is ACP means alcohol induced KC mice. We saw tumor. You see the tumor here. This is the pancreas, completely pancreas, and we saw the tumor. And we were curious at that time to develop more mice and to study more in depth and understand the biology of it, right? And then we went ahead and did in the KRAS model. This is the KRAS model, like the same model I showed you without the KRAS. Now you see the KRAS. What happened here? In the 3-day, we stopped everything. We gave 3 weeks of alcohol, and we stopped. Inflammation is happened, and now which has a KRAS. Now you see all the tumor cells are coming out. Means that when you have a KRAS, the ductal cells are all not going back, not reversing. It's continuing, and you see more of ACNR cell, Alcian blue stains for all the ductal cells, and there is a lot of fibrous. And if you see the histology, you see a lot of cancer. We measure all those things, and then we analyzed here. You see a lot of cancer within 21 days. These are all pannins, also kind of a cancer. Okay. Is overall the KC mice with alcohol inflammation induced accelerated this pannin and cancer development. So this is just to show that we compared this without KRAS and with KRAS, and without KRAS with CP means that just the inflammation, no alcohol. Just I want to have just the inflammation, no alcohol. Okay, it's a drug-induced inflammation, no alcohol. When compared to just alcohol adding to the inflammation is even bad. I told you just now that these people keep drinking and they stop, again drink, but even though there is a pain, they keep drinking, even they think that the pain will go away. Exactly what the model is here. They have inflammation and they keep drinking, adding alcohol actually is worsening. They won't know. Your body cannot see inside, only you see outside. Right? Inside what's happening, only you can feel it. You cannot see it. Right? And unless you use the technology. Now we have a technology. So in the KRAS mutant mice, same thing if you compare, same age group, same everything, all these mice, now you see the lot of tumor in the KRAS mutant one. This was very interesting for us, but as a biologist, you know what I'm saying? What are you going to do? I want to know more. Because that is the thing. I want to know more. So to know more, what are we going to do? We do single cell sequencing. You know what is single cell sequencing? You will know more next upcoming days or even later on that just a cell, one tumor cell is not enough today. You have to understand different cell types, how they work, how they interact each other, how they communicate each other. Okay? For example, each B cells, T cells, ductal cells, CAF cells, and neutrophils, and all these macrophages. You have to understand all these cells. That's why you do the single cell sequencing, so that you will know all these cells types. And then what we saw here, we analyzed all these numbers, ACNR cells, control cells with the ACP. You see ACNR cells are going down, and in the ductal cells are going up. What does that mean? Means that ductal cells are increasing. Means that cancer cells are coming out, and ACNR cells are going away, normal cells are going away. If you don't have a normal cell in your body, if any damage happens, you will not be able to recover. We all know. That's why when you get aged, your inflammation, it's very hard to control, right? We all have our grandparents and all. When they have inflammation and everything, it's very hard. Because these cells, they don't have normal cells anymore. They're all aged cells. So these cells cannot recover these things. That's why we have to take care of them better, right? Because they don't have those normal cells. We have a normal cells. Our kids can break it. They break the leg, they come back. They break the finger, they come back, because the cells are so strong. They only have a normal cell. They don't have any other dirty cells, right? But as we middle age, we have a problem. Recovery is a little bit slow. As we get older, it's slow. It's exactly the same thing here. Now you're drinking alcohol and you're reducing your normal cells. You're reducing your normal cells. If you get infected with something else, contamination of something else, like someone is drinking alcohol and he is maybe having a pesticide somewhere. Just imagine the problem to this person. Something might happen, right? So something like that. So it's just a thought. So these are all the biology. I just don't want to go in detail of this, and the person who studied those, technology. See, each technology I'm going to show, there is a person who actually do in the laboratory, or in the neighbor laboratory, or the friend laboratory, colleagues in different departments, and they conduct those experiments and give it to us. So it's an interactive program. It doesn't matter what. When you're studying, you have to have your colleagues. You cannot close your door and study biology today. So you have to have the technology and you're not yourself. You will not be a scientist. You are binding to other people to conduct those other experiments. So you need more friends today. So here I want to show you these confocal images. I'm going to show you. I'm showing you here is that this is a 3-day. Okay, this is a 21 day in a KRAS mutant mice. What you're seeing here is that beautifully, okay, in these mice, that the green is going away and the red is coming back. Means that the ACNR cells are going away slowly. You can see some cells are going away, some cells are off, some cells are completely clean ACNR, but some cells are completely ductal, right? They're all changing. I'm not lying, this is true. This is exactly what you need to understand. It's a curiosity again. Our curiosity is going, see, when you see it, it is there. You have to investigate. When you see it, don't leave it. There is something. Always go back and study. There is always something. But how you develop that curiosity in your hand. It's in your hand. It's all about your time and your effort. If you have time, if you are interested, it will happen. If you are not interested, it will not happen. So the same way, we went back and you know what? I'm not going to stop this. I want to understand this more and more, how this is happening, why this is happening. I want to know beautifully. One picture can tell you the story. Same thing if I put in a blot here, people can make it. But you know what picture? I cannot make it. You know what I'm saying? So this is happening picture. That's why in my study, more of picture you see because I want to know what is happening rather than just giving data. I'm not curious. I want to know. In my eye, I want to see it. Okay? Single cell, I see it. I see T cells, what's happening. I see B cells, I am curious. If you take the spatial genomics today, it's happening. You know what? The genomics, when you are spatial genomics, it just takes the area which you select, and you tell them based on the histology, based on the many factors, markers you study, and you tell the technologies, okay, you pick this area and tell me what genes are changing here. I want to know. Then they will give you. That's why this technology is so beautiful. But if you cannot do it, it's also beautiful. You know what? How much ever you can. You don't have to crack your head, okay, I don't have a genomic study, I will stop my research. No. You will never stop research. You have to keep doing the research, but whatever you can do, right? That way. This is one thing. So another way we wanted to know, in just a simple qPCR analysis, taking all these genes, ACNR genes and the ductal genes, there are qPCR arrays you can run, right? There are arrays, you can actually design your own array today. So there were days that we will not be able to. Today you can design what are the genes you want to. For that, to select those genes, you have to do research. You have to collect a lot of information and then put it together, right? And then we analyze the tumor latency. What does that mean? The tumor latency analysis is that we put a lot of 10, 15 mice together and we do the control alcohol, chronic pancreatitis and alcoholic chronic pancreatitis mice. And alcoholic chronic pancreatitis mice, they die faster. They make tumor faster when compared to just alcohol. Just if you do the alcohol, maybe they may develop tumor or may not develop tumor or tumor-like ductal cells, but they will not become cancer cells. They go back, right? And the chronic inflammation, they go back. The alcoholic chronic pancreatitis is actually pancreatic one is actually one. With the KRAS I'm talking about, with the KRAS it will go further. Without KRAS I'm talking about, they go back. With the KRAS, they go forward. With the KRAS I'm talking about tumor latency, that means that ACP develop the tumor faster, right? So now, all done. Now what is important for us as a cell biologist, for me, understanding the signaling. We are curious. As a cellular biologist, we want to know the signaling. Joshi also wants to know the signaling. He works on the F1 alpha. And someone else, he was actually curious on F1 alpha. I was asking him, why F1 alpha? But we have different ways of thinking, right? Our ways are different and his ways are different. Like that, every researcher think differently. So you think differently. You may think that, oh, EGFR I will study. Someone will say, I want to study maybe AKT. Okay, yeah, go ahead and study. And tell me why. The reason you have to give me why. Important, right? Okay, there are tons of people who have done already AKT. Why you are doing also? You have to tell me the story though. We have done AKT, but we know the reason why AKT, right? It's important. So then we did the kinase array, means all the kinases, we did the array, and we selected the top kinases that changed, and one of the kinase that we selected is the CREB, because I already was studying on the CREB, and also that popped up here, and we saw that with the alcohol, the CREB was going up, and it was going down again. Means that it is activated when it is damaged, and it's going back when it is recovered, right? And we did that study, different models, different ways, and then in KC mice, in the KRAS mutant mice, we saw the expression of CREB beautifully. Look at that. The CREB is expressing, that is the CREB is in the green, is here, and expressing on the ductal cells nicely. Wherever the ducts are there, the CREB is there. So it is like you see in your own eye that it is working. It's expressing. Means that you know that it is working. So that is very important. Important is that picture. Seeing yourself that this is working is a key thing, key factor. So picture perfect, right? You say, oh, this is a picture perfect. That's how it should be. The CREB is activated. You see here, there is no CREB here. When you have a hit with alcohol, we see the CREB. Now we got confirmation. And now we took all the pancreas tissues. These are all tissue analysis. And we took like three mice of each study just to run the protein of the phosphorylated CREB. We saw, with a 3-day, a 21-day, a 42-day, means that CREB is not going back. What does that mean? Means that the KRAS is activated. The CREB is a cyclic AMP responsive binding factor, is one of the transcription regulator. If you study molecular biology, the CREB and all these things will come in a, CREB is one of the standard master regulator. So we can talk about CREB later, but important is not CREB. Important is that you think like, there is one target is activated. Okay, think like that. It can be something else in a different cancer, right? Maybe somewhere in AKT, somewhere in the PI3 kinase, somewhere in STAT3, somewhere in some other model, maybe in other model, maybe something else, right? So this way. Here, we were even more curious. Can we do some mice model? Can we add something like in a color through the mice? You know what I'm saying? So that we will see exactly what is happening to those cells. So we added this, green color to these mice. Means that another mice we added attached to this, and with a green color. And this, wherever this Cre, means that wherever the pancreas cells are there, acinar cells, you see the green there. Okay? These are all this natural. We are not put a color or anything. It is a natural mice model where wherever the acinar cells are there are green color. Okay? The same way today you can make tomato color, you can make different colors of your different. So it is to study, it is to understand the biology of it. It's a technology developed. So we have to use it though. You have to use it and study. So we use that mice, and if you see here with the YFP, that is YF means a green color, and the CK19 is the ductal cells, and the phosphocreb is our red color. We added these two colors. This is the natural color, and wherever the YFP is there is a CREB is there. What does that mean? Means YFP is changing to blue. Means that green is changing to blue. You see the blue colors here. That means that green is changing. The green is going away. That means these mice cannot hold the green anymore because it's damaged. The acinar is gone. Normal cells are gone. Now they're all ductal cells, right? That's why you see those cells changing. So that was curious. We were curious, and that happened. And just to show the model that the CREB is activated and
keep going, means that we were now interested in crab, okay, even more interested in understanding the crab and the biology of the crab, where it is going, how it is happening, and where is this crab binding, why only crab, why not something else, that means that what else is crab doing, things like that. So this is maybe a one model. Remember, for you, it's just concept ideas, okay? It's maybe a crab for me, maybe something else for you. But you have to think, it's just a concept idea. When you get that molecule and the model running, right, you have to think what else you can do. And then we created the crab knockout mice. That's what we do as a biologist. What we do, okay, now crab is activated, take that off. See what happens. Delete the crab. And then if it is crab, that means that if it is deleted, what will happen? It should not happen, right? Cancer should not come if it is crab mediated, right? Then what we'll do, if that is true, we develop the drug to the crab and then we treat this patient. This is how we do to the treatment. This is how you develop the model, you develop the concept, you develop the idea, and you model that idea in the research, and then you model the mice and study, understand, and then you go back and try to develop the genetic mouse model and delete that gene and study, and then if that is true, because genetic model, when you study, it's true, that gene you deleted it, right? It's working, and now you target, you develop the drug, and next comes is okay, crab has a drug or no? We don't know. We'll come to that. We will talk more on that. Okay. And then what we did when we deleted the crab here, we studied and making sure that the acinar clusters, and when the crab is deleted, all the ducts were not forming. The acinar cells are acinar cells. There was no ducts. That means that crab is working. So when you delete crab, with the KRAS mutation, there are no ducts. Means that it is acinar to ductal reprogramming. Means that it is reversing the process by deleting the crab. Okay. And also just to make sure, when we deleted the crab and whether we have more acinar cells, okay, we lost the crab duct cells and there are more acinar cells. What does that mean? We are also trying to understand in the transcriptionally gene level that okay, we still have these normal cells in the KRAS mutant mice model. In the KRAS mutant mice model, what happened? acinar cells went into duct cells, right? Now you deleted crab, and now that there are no duct cells, there are still acinar cells, even though there is a KRAS, right? The crab was mediated in this process. Means that crab is really working. That's why crab was popped up when we did the kinase array assay. Remember, in the research, it is not just one-time experiment. Only you are seeing years of experience in one slide, crab is activated. But to get the data, you have to struggle. It is easy, one slide, one data, but it is not that easy, right? When you do multiple mice, there is a multiple way of data you get. So you as a scientist, finalize yourself that what data you're going to take that and move forward. Based on many factors, that is research. Again, it's not something that, okay, oh yeah, I took it and moved forward and it worked out. Probably in the movie, not in research, right? So it may not work that way, but you have to, as a scientist, sometimes you have to analyze yourself, think yourself, develop some thought process, parallel thinking, okay? So if you take there were other kinases I did not take. You can ask me, why you didn't take the other one? Because I want to take something new. I want to study something new. Maybe there is AKT also doing the same thing. There is something STAT3 also maybe doing the same thing. I just don't know. But STAT3 is well known. AKT is well known. CREB is not at that time. Now it is well known because we published it. But what I'm saying is that maybe STAT3 is doing. You may ask me, okay, that means the drug is working. How? Strategy was also activated, AKT is also activated. Maybe they are still activated. Yeah, they may be activated. But this is also working. That also may work. Okay? Then, for example, if you take a disease, there are many multiple drugs. Okay, if you take a fever, there are multiple drugs, multiple compounds work on the fever, but multiple targets. Remember, but it actually takes care of the fever. The same way, here, it takes care of the models and the study. It's the same thing, same concept, same design. You can ask me many questions, but as a scientist, you have to have analysis thinking. Analyze it, put a common factor, and design your own science and ideas and science, things like that. So I just don't know how much time I have. Yeah, we can continue afterwards. Okay. So let me stop here. I know that you're all hungry. We can continue afterwards. But what I wanted to show you next is we are deleting the crab. Okay, say in my part is a crab, maybe for you something else also. Think logically in your research or the way, or maybe if you wanted to do something research in your own model, think logically. If something crab kind of a thing comes to in your lab, some other molecule, how you want to study, why you want to study, the same concept, same design, curiosity, develop interest, and design your model. That's all I'm trying to tell you. So maybe after lunch we continue this, and I'll tell you more story about it. Remember, I'm taking a pancreas as an example. I'm giving you a hint how you want to study because it's easy. For me also easy, and for you also to understand with the data, with the our own realistic ideas, how we developed, probably will help you. So all our, most of our talks every day, same concept, same design. Thank you.